Immune Memory and the Coronavirus, Part 2. How do cells remember? When you've recovered from an infection or received a vaccination for a particular pathogen, like a virus or bacterium, you will generally develop protective immunity specific to that threat. However, the strength of the immune response, how much protection it offers, and how long it lasts varies with different diseases and vaccines, and from person to person. So when it comes to preventing reinfection, how exactly do our body's cells remember? In the previous video, we looked at how the immune system functions and what happens on a cellular level when a person responds to a viral threat, like the coronavirus. We learned about the innate and the adaptive immune responses. B cells and T cells, which are part of the adaptive response, are trained and selected and then make clones of themselves to neutralize the threat. These cells respond by making antibodies or by killing infected cells. Keeping these specially selected B and T cells in the body is what helps us remember how to fight off a specific infection if we see it again. This is immune memory. After selection, some of the active B cells will change into memory cells, which circulate in the body, including near the site of infection. Memory B cells live for much longer than untrained or active B cells. They are selected over several months for the best possible fit for the target virus or bacterium to provide better protection if reactivated. Memory B cells do not keep making antibodies all the time, but if the original threat returns, they can quickly be reactivated to make more antibodies and more clones of themselves. Most people who've had COVID-19 do have antibodies against this coronavirus. But what antibody characteristics help you survive the disease, and how long antibody protection lasts following infection or vaccination, are questions that are still being studied by immunologists. While B cells primarily make antibodies, T cell functions in the immune system include activating other immune cells and killing infected cells. After formation of a protective immune response, different kinds of memory T cells can also be found in tissues and circulating in the body. Immunologists are still trying to unravel the puzzle of immune memory roles for the different types of T cells, which have been shown to be important in protection against many diseases, even when antibodies can't be detected. Current evidence shows that during infection with or vaccination against the virus that causes COVID-19, people develop a strong T cell response. However, this can sometimes be less robust in older people. The most crucial part of immune memory is what happens if a person encounters the infection again. The much faster speed of this secondary adaptive response, once you have a protective immune memory, normally means that you are much less likely to get infected and ill again. Unfortunately, the adaptive response to some infections does not create effective protection. In other cases, it actually has overlap and can provide additional protection for similar pathogens. You can learn more about this in our other videos. Immune memory and the fast efficiency of the secondary immune response is the reason vaccines have changed the way we live. By showing parts of a pathogen or a weakened version of it to the immune system, a vaccine can train the immune system and create a protective memory without causing the illness. This means that the immune system knows what to do without having had the risk of a person getting ill in the first place. It provides safe training for our immune cells, giving time to select and retain memory T and B cells before they face the threat for real. Much like the emergency services responding to an outbreak of fire in different settings, the immune system will tackle each threat it faces in a slightly different way. This dynamic and sometimes complex balance of innate and adaptive and T and B cell responses needed to guard against a specific illness is known technically as the correlates of protection, or COP, for that illness. It's what part of the response actually works effectively to protect you. To know how best to treat and prevent an illness, immunologists have first to understand these correlates of protection, working out what the body does naturally in an ideal response. This research underpins the development of successful vaccines, as well as reliable tests for protective immunity after infection or vaccination. Currently, a significant minority of people with COVID-19 show little or no detectable antibody levels after testing positive for the virus, but this may be due to mild infections or to the effect of old age on the immune system. Thankfully, early reinfections appear to be rare. If immunity is largely achieved by B cells, then antibody testing ought to be a good correlate of protection. Different tests are needed to examine the T cell response. T and B cell responses appear to go hand in hand for COVID-19. The new vaccines seem to generate T and B cell responses with protective immunity. The ultimate goal is to produce a vaccine which provides long-lasting protection against both severe disease in individuals and asymptomatic viral spread in the community. The cost, storage, and number of required doses of a vaccine also contribute to its usefulness on a global scale. Current coronavirus vaccines achieve a very high rate of protection with 1 or 2 doses. Ongoing research will help us to reach the best combination of vaccines and booster jabs in different groups and different settings. We will discuss how long immune memory lasts in Part 3. In this short video, we've learned about the basics of immune memory and how a secondary immune response is more rapid and efficient than the initial one. We've also learned about the importance of understanding immune memory and the correlates of protection for each individual illness. This research helps to create more reliable tests, vaccines, and treatments. If you'd like to learn more about what happens during an antibody test, the role of individual immune cells, or how vaccines are made, please use the links below. This video is part of a collaboration between the Centre for Inflammation Research at the University of Edinburgh and the British Society for Immunology. Thank you for watching.